Now I've got a fair amount to say, so I'm going to go quickly through a bunch of slides. So bear with me, but I want to motivate. Let's see, how do I actually press the button? There we go. Yeah. I want to motivate the discussion because in the title it says reflections on biodiversity. And of course, the reason I want to reflect on biodiversity is because of these guys who we just lost over the last Christmas, Ed Wilson and of course, Tom Lovejoy. Both of them champions of biodiversity, bringing it to the attention of the world really, and doing so much to help solve the problem while they could. But Ed Wilson, many of you will know, has been associated with Yale in various ways. He got an honorary degree here in 1999, I think it was, either 98 or 99. He received the Verrill Medal from the Peabody Museum. He consulted in various ways with several of us at various times, including Ed Bass in the early days of setting up IPBES. I think some conversations with Ed Wilson were really formative in some ways. And so he's been connected, although he's been at Harvard was at Harvard his whole career, very connected here. But Tom Lovejoy, of course, was here pretty much on a daily basis. It seemed like, he was with us all the time, and really contributing really brilliant ideas in a very calm way that he does, but very calm and thoughtful and quiet. But boy, did he have a lot to contribute around here to IPBES and the Peabody. And I miss him a lot. Anyway, it seems like a good time to reflect on biodiversity in the wake of that. I'm having trouble advancing the slides here for reasons I don't understand. Oops. I'm sorry, but it's not working. Oh, I don't know. I hope I can get that to work regularly, but because I don't know what I did just now. Well, listen, I have three parts to the talk, and I'll go through them, expeditiously as I can, I guess. The first part, I want to think about refocusing attention on the loss of biodiversity, because I think it's kind of lost a little of the edge that it had in the heyday with Wilson and Lovejoy bringing attention to it. And then I want to talk about using evolutionary history, which is sort of my specialty, to sort of measure and value biodiversity, just to bring that to your attention, that there are ways to integrate the kind of work I do on phylogeny with biodiversity conservation. And then I want to apply what we learned from that to what's going on in this group of plants that I worked on called Viburnum. And I'm fascinated by these things. I know they're fairly boring to other people, so I'll gloss over a lot of that. But okay, so reflecting on what's going on with biodiversity, it seems to me, and I'll be blunt about this, that climate change gets a lot of attention, as it should. I mean, climate change is a big deal, and we should all be aware of it and doing the best we can to, combat climate change and so forth. But in the mix, biodiversity and the biodiversity crisis has kind of taken a little bit of a back seat, I would say, in terms of the way most people perceive problems in the world. The first thing they would think about is climate. And secondly, maybe think about biodiversity as a crisis. Now, what's the reason for that? Well, one reason is since a long time ago, 1988, actually, like 25 years or something, there's been this thing called the IPCC, and everybody in the room is familiar with the Intergovernmental Panel on Climate Change, because they put out these assessments from time to time that say, here's what's going on in the world, here's what we're predicting in the future, and so forth. And these are very, very well done, and they're well broadcast, I guess you would say. So that's been a big plus for the climate change community. The other thing about climate change, of course, is that things just sort of knock us over the head, things like hurricanes and wildfires and stuff that can be attributed, I think rightly, to climate change, or at least exacerbated by climate change. And you notice these things. I mean, they're in the news and they happen overnight. Suddenly there's, Hurricane Harvey or something is hitting, and you can't ignore that sort of thing. So people are connecting the dots between things that are affecting them very immediately and global warming. So I think that's been an easier sell for people than biodiversity, which is a little more abstract in a way. Now, biodiversity science has its IPCC equivalent, but only since 2012. So it's only been, 10 years now. And this is called the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, or IPBES is the way it's said in the community. This slide in the middle shows something just from the homepage right now, saying that North Macedonia just joined as the 139th member state So there are a bunch of governments involved in this thing, and they meet regularly, and they try to do global assessments. So far, they put out one major global assessment, this one in 2019, and it had it was an impressive document, really super impressive and scary in many ways. And it did catch the attention of the news media, at least for a few days as it goes. But the main thing that caught on in the media, at least, was this thing, because there was the prediction of the loss of a million species over the next 2 decades. That's a lot of species over 2 decades out of a total of 8 million maybe on Earth. That's that's questionable, but something like that. So that's a lot. Could stand to lose a lot of species over a very short time period. And this is a graph from that report that shows that for various groups of organisms, there's some number that are threatened or vulnerable or on the verge of extinction. That's the ones that are sort of in the red colors. So for cycads, which have been very thoroughly studied, about more than 60% of them are endangered, potentially going to go extinct, and so forth. So amphibians, it's something like 42%, something like that. So you can see that blue line is kind of representing the numbers that are endangered and so forth. So it's really problematical when you look at that and you think what could happen in the future. So biodiversity loss is a big deal. The other thing is that it's not just about extinctions, it's all sorts of other troubling signs, like population sizes are declining all over the place. Like this very impressive study of birds in North America shows the reduction of the number of birds by 3 million over the last 40 years. This is the sort of thing that's going on, but very quietly and slowly in some sense, compared to our daily lives. So we tend not to notice it. It's quiet. You don't really feel it until it's kind of too late or something like that. So it's not like the hurricane or the wildfire, right? So it's a very different kettle of fish, really. And it's a much harder sell for people. Now, unfortunately, the way people have sold the biodiversity crisis, to a large extent, I think misses the mark. And this is, I'm not alone in this view, but maybe not the majority either. So I'm just going to express my views and not attribute them to anybody else. But basically, I think we've sort of taken the wrong tack when it comes to biodiversity and highlighting it for people in terms that they can understand. Because what happened was, starting, with the Millennium Ecosystem Assessment, which is a global assessment in 2005, which is a very impressive study of the world's ecosystems. Starting from that time onward, biodiversity was sold, in a sense, to the community, to people, as a problem with ecosystem services. It was associated with ecosystem services. And what are ecosystem services? It's all the things that biological things that the biosphere does for us, like regulate water and regulate erosion and photosynthesis and nutrient cycling. It's everything that's on this chart, which is like, it's a big, large, important number of things, but it's pretty hard to get your brain around all of this. And it's pretty hard to associate biodiversity loss, like the loss of some cycad or something, with any of that, right? In other words, how would that affect global photosynthesis? How would that affect erosion or something? So the connection between biodiversity, like losing whole genetic lineages and such like that, and these things is a bit abstract for people. It's very hard to pin down. It's hard to pin down even for the best scientists who study this stuff, much less for the public, right? So I think we sort of made a wrong turn there. And I mean, I had something to do with that, so I'm partially responsible, but I'm taking it back now. And but I do think there's parts of this that we could focus on to very good effect with the public, and it would be more like the hurricane or something, or the wildfire. And the one that when I'm asked to give talks that relate to biodiversity, I often focus on food examples, because everybody loves food. What would we do without it? It's very clear that it's very important to us, right? And there's a very clear connection between food and biodiversity, which I'll make for you with a couple examples, okay? So I like to start with something like this. It's like, if you think about it, we're just, we're inundated with biodiversity every time we go to the store. I don't go to the store often enough, but when I go to like the Stop and Shop or something, I'm literally floored by it. I just can't believe all the food that we have in there and all the different types of food and everything. And then if you go to an Indian market or an Asian market, oh my God, the diversity of things that are in those markets, it's unbelievable. But this is something that people experience, and they kind of, it sort of washes over them. They don't sort of connect this with biodiversity in some ways, but there it is, biodiversity, right? So, I think it's effective. It could be effective to try to connect biodiversity to food and the kinds of things that people like to eat. So I'll give some examples, 2 examples actually. One is, and I'll go into a little depth about each one so you can get the flavor of it. But this one's about coffee. So what's up with coffee? Well, everybody loves coffee, I think, most people. And there's the Arabica type coffee, which is about 60% of the market. And there's the Robusta type coffee. And Arabica is the better of the two in terms of the flavor and everything. And so, for example, Starbucks will only sell Arabica. They make a big deal out of that. These are the countries where coffee is grown. The green is where Arabica is grown pretty exclusively. The red is where Robusta is grown. So this thing from Vietnam is coming from a red country over here. So there's Robusta coffee. These are two different species of coffee and Arabica. And that makes up 99.9% of the market. There's very few other coffee varieties or coffee species that are ever used. It's a huge industry. I mean, this is gigantic. Many people rely, are buying coffee. Many people are producing coffee. Many people are selling coffee. It's just monster business in a way. Okay, so a little bit about coffee. So it's a shrub. It's a plant. It has flowers that look like that. It has fruits that look like this, little cherry-like things. They're sort of called cherries in a way. You open it up, and inside of it, there's 2 seeds, and the seeds are surrounded by some little stuff. It doesn't really matter to us. There's 2 seeds in there. And then what you do is you pick them, and then you dry them out like this, and then you mill them a little bit to get rid of some of the crud, and then you end up with these seeds. And these seeds then get roasted to varying degrees to make the coffee that we like, right? So that's basically the coffee situation. Coffee, the one that we like, Arabica, is a tetraploid, meaning it has twice the number of chromosomes as any other coffee species. It originated as a hybrid between these 2 other ones. Robusta was one of its parents, in fact. But these are sort of details. But many crop plants have this kind of origin, something like this going on with them. When they were brought into cultivation, they were often a little bit odd genetically. Well, coffee plantations are now, kind of being devastated by several things. One is there's a rust called the coffee leaf rust, which you can see there. That's a horrible thing for people growing coffee. And the other thing is this little beetle here called the coffee berry borer. And it bores little holes in the fruits like this and goes in and eats the seeds. That's obviously bad for coffee growers. And the other thing that's happening is climate change. So global warming is forcing the movement of these Arabica zone, the zone where we could cultivate it, which is basically above 600 meters. As climate is warming, you have to move up the mountain, basically. And as you move up the mountain, you run into all sorts of problems with other things that are happening up the mountain. So it's kind of constricting the zone where you can grow Arabica coffee. So that's a problem. Now, it turns out that like all of our crop plants or animals, they have wild relatives. And in the case of coffee, there's about 125 other species of coffee, right? But we don't know very much about them, but they look different. Some of them look kind of different. I mean, this is sort of regular coffee that we're familiar with. Some of them have different kinds of colors of fruits and things like that. But there's a bunch of species. Now, this is a phylogeny, an evolutionary tree, and I'm going to say more about that in a few minutes. But what I want you to see is that there's a part of it that's kind of yellow and green like this. This is the part of the coffee phylogeny that has caffeine. The other part doesn't have caffeine, so who cares about those in some sense, right? But anyway, these are the ones we would want to concentrate on if we're thinking about coffee. And I in a circle here, I put this one here, Stenophila, which I'm going to mention to you in a minute. Okay, so that's a little bit of introduction to coffee. So there's multiple wild species. Now, it turns out that these wild species, there was a paper just published just recently that showed that many of them, about 60% of them, are threatened with extinction. So they have very small populations. They're just in very restricted part of some country in Africa, let's say. And so that's the ones that are in those kind of red colors up there. Those are the IUCN sort of red list, right? And then some of them are not threatened, but many of them are. The other thing is if you go and do a Wikipedia search, right? This is what you get on the coffee page. And as you know from Googling around and everything, if it's blue, you can touch it and go on and get more information. If it's red, there's no other information, so that's the end of the line. So the bottom line is we don't know anything much about any of these coffee species. I mean, just a few of them are blue. Well, one of the ones that is blue now, but wasn't a year ago, is this one, Stenophila, which is the one I'm going to mention to you just now. So what is that Stenophila? So this is the distribution of coffee. So it turns out Arabica coffee is that blue thing right there. It comes from Ethiopia. It was originally cultivated there and also in Yemen there across the way. And that's where coffee became popular actually as a drink. And this is the Robusta coffee is in this zone, right? But then over there, there's this other thing, and that's the Stenophila. So that one that's over there in Sierra Leone and the Ivory Coast, that is an interesting one. It was first discovered long ago, 1794, described in 1834, and then it went under the radar somewhere and nobody saw this plant for a long time. The last time it was seen was in 1954 until 2019 when some people found it again. One population in Sierra Leone, and then they found another one in the Ivory Coast. Just a few plants. And it turns out it's a pretty interesting species. For one thing, it has black fruits instead of red ones, and it has skinny little narrow leaves and everything. So it's distinctive as a coffee plant and everything. But what about the taste of it? So people wondered, would this thing be any good to drink? So you take some coffee beans and you put them into cultivation, and this was done in the island of Reunion. And you grow out a grove of these things, and you harvest them, you make coffee out of it, and then you serve it up to the best coffee people in the world and sort of say, what do you think of the flavor? And they have, it turns out, a kind of a way to kind of judge coffee flavors. And it's kind of a science and an art. I don't know what it is exactly, but they measure fruitiness, acidity, body, and all that. Now the red line there is Arabica coffee. And the one that's almost coincident with it is this Stenophila. So Stenophila tastes like Arabica coffee, which is great, right? So it could be used as coffee and people would buy it and they'd pay money for it. But the great thing about it is it grows at much higher temperatures. It grows at the temperatures that Robusta coffee. So you can grow it at sea level, basically. It doesn't have to be up in the mountains. And the other thing is it has no pests. None of these pests. So it doesn't have the rust, doesn't have the beetle. So potentially this could be integrated into the coffee industry here as a solution to some of these problems. If not replace Arabica coffee, it could actually, be bred with Arabica and ways to deal with these diseases and stuff. So in a sense, right under our nose was this potential solution to a giant problem that's looming. It's this thing that was growing in Sierra Leone. And now we know, right? But of course, we could have lost the thing altogether, right? Bummer. Then we wouldn't have that solution potential, right? So that's an example of a food problem where I think biodiversity comes in. And people can relate that, oh yeah, we want to preserve biodiversity. Second example has to do with corn, and I think everybody realizes humanity is totally dependent on corn. And so it's a big deal. Now, the thing I want to focus on here is nitrogen fertilization, which we do all the time in Michigan and other such places. And a lot of that nitrogen runs off and goes into the Gulf of Mexico. And then we get these big dead zones there where nothing can live because the oxygen plummets and everything. It's just a big, awful mess. So that's a problem. So growing corn is problematical from the pollution standpoint. Now, there is, it turns out, as this has just been discovered in the last few years, a special race of corn that's cultivated in Mexico in this area called the Sierra Mixe, which is where, and the center of that is a town called Totontepec. And this is a picture, basically from Totontepec up at the mountain that's nearby. I know this well because I took that picture because, as I'll show you later, we work in that area all the time. And so I've wandered these cornfields as I go up to the mountain to find my plants. I go through the cornfield. And this is what the corn looks like. It's really great, robust, really tall corn plants growing really well on these really poor soils. And there's no nitrogen in the soils, and they're not fertilized. So people, when they go there, they go, how can you grow such wonderful corn without nitrogen fertilizer on this kind of soil? Well, so now I have to tell you, normal corn has prop roots. You've seen that, right? Everybody's been to the Midwest or something somewhere and seen these prop roots. And it helps hold the corn plant up, and the roots are in the ground and everything. Well, it turns out that this corn variety from Totontepec, it has these things at every leaf juncture all the way up through the plant, basically. That's what's shown here. So normal corn just has a few of these at the base. This is going all the way up the plant, right? So every node has a bunch of adventitious roots coming out. So what's up with that? That's weird. And the second thing is, can you see it's kind of covered with some sort of slime? It's like dripping off of that. So people are going, what's with these prop roots? They're not propping up the plant. What are they doing? And they're covered with slime. So people started to investigate that just over the last few years. And the first paper on this was published in 2018, by the way. There's been a flurry since then. But basically what was discovered is that in that mucilage, there's a whole bunch of complex polysaccharides that are being broken down by bacteria that are anaerobic bacteria, a whole microbiome living in this gooey stuff. And what they're doing is fixing nitrogen out of the atmosphere. And then the plant is soaking up the nitrogen, right? So basically it would be the equivalent of a root nodule in a legume, like alfalfa plant or something like that. Nitrogen fixation is happening in this place in here, and these plants are thriving without nitrogen fertilizer. because of that. Isn't that amazing? So anyway, there was this was the first analysis of this, showing that there's all these sugars in there. What are the sugars doing, and so forth. Now, since then, there's been a bunch of studies, and we know a lot about all the reactions that are taking place. You have to break down the polysaccharides. The bacteria can only operate at essentially no oxygen. They're anaerobic. So you have to have enough of this gooey liquid around so that in the middle of it, there's no oxygen. That's a key component. So the question is, how can we get this to happen in other normal corn plants, but how could we get it to happen in other cereal crops, let's say? And so the idea is that many plants make a mucilage, but if you could just make a whole lot more of it in certain places, you could start to grow this microbiome and then solve the fertilization problem. That's cool, I think. So another example of, hey, it pays to preserve biodiversity. I mean, this is a really wonderful case of something that's going to sort of save our butts in some ways when it comes to this pollution problem. Now, this has some other sides to it, and this was reported in the Yale Environment 360 publication, in fact. The question is, who owns this? Who owns the rights to this wonder plant? You know, and that's been a really big, complicated issue with this particular case, because all of the villages around Totontepec were not involved in the actual negotiations over who would share the benefits. So there's a lot of consternation in that part of the country now about, like, wait, is it only Totontepec that's going to get any sort of benefit out of this, or does my little village also, you know, that kind of thing. So it's very interesting case study about benefits sharing. Okay, so I'm done with part one, basically, and all I've said is that I don't think this ecosystem services thing is going to work. I think it's too abstract. But I think these kinds of examples that sort of bring in, you know, real life things like coffee and corn and stuff like that, I think they can really be powerful. At least I find that when I go around and talk about this stuff. Now, I want to talk a little bit now, segue into part two, to talk about my own involvement in biodiversity conservation community, so to speak. And I say this, but my involvement has not been so great, but I have had some, and I want to highlight one or two things from it. I was involved in a program called Diversitas, which was the first of these large international programs that was about biodiversity. And eventually it spawned IPBES, the thing that I told you about, right? And so I was kind of co-chairing that thing for about 7 years. And as one of the things I did, within that Diversitas program was create this sub-program called Biogenesis, which was focused on, as it says there, evolutionary framework for biodiversity science. And I thought that's something I can bring to the table that other people maybe can't bring. So I worked pretty hard on this for some years, and we published a science plan. That's what that is right there. And then a series of other publications came out about, you know, that how could we relate phylogeny and evolutionary history and the kind of thing that I do, how could we relate that to biodiversity conservation? And so that's had some effect in the community. In fact, I think nowadays most people doing biodiversity conservation will have this kind of perspective, you know, baked in to some extent. Now I need to tell you about phylogeny in a little more detail. So the way I do this or whatever is with fingers initially. So, you know, there are various symbols that we make with our fingers. You can make with one finger, you can make some. With two, you can make some like that. What is that? That's like peace sign, right? I mean, most people recognize that's the international peace sign. So if you just do that and then take your thumb and put it out like that, that is the international symbol for phylogeny. so that's a little phylogenetic tree, right? So you can actually, you know, if you start with any finger right here, and imagine the tips of your finger are these species at the tips, they're these existing species. So I go down from this species here and I travel down my finger, and what that means is I'm going back in time. So this is the lineage leading up to this species at the tip of my finger, right? Now, if I go back in time far enough, I hit this place where I'm intersecting with this other finger, right? So I've gone back a certain amount of time. That could be measured in actual absolute years. Let's say it's 2 million years before I hit this ancestor, and then there's the other guy coming off right here. So this was an ancestor that split into two species like this. And then if I go back further, I eventually hit this intersection with a third species over here. But that one has an ancestor that's back further in time. Does that make sense? So you can explain all of phylogeny just with like that, right, if you do it right. So now, just to see that you've got the concept, here's a test, I guess. So this is actually from the publication that first defined phylogenetic relationship. It was a German publication in 1931 by a botanist, it happens, Walter Zimmermann. And he's the guy who's defined what we now take to be phylogenetic relationship. So which of these two species is most closely related? Of these three, which two are more closely related to each other? It's so obvious, right? So what would it be? The magnolia and the applebaum, the apple tree, right, are more closely related than either is to the ginkgo. Is everybody agreeing with that? Okay, and it's important to get straight on why. So those two are more closely related, but why? It's because they share a more recent common ancestor. So there's this common ancestor at X2 here that's more recent than this one. So to get to the ginkgo, you have to go back further in time to hit an ancestor that they share, right? So that's the notion of phylogenetic relationship. And then you can just keep expanding it. Like if I have this, I can just add this, or I could add this and so forth. And eventually you go all the way back through all the tree of life, through all of the nodes way back in time. And then you've got the whole thing. That's the phylogeny of life, right? Are you with me? Okay, so that's just introduction to that. Now that allows us to take a step in the direction of this biodiversity conservation thing. So there are ways in which we can calculate how much we value various species from this standpoint, actually. And there's a program called the EDGE program, which stands for Evolutionary, Distinct, and Globally Endangered. So the idea is to try to identify species that are these kind of special ones that stand out. And in this particular case, it's this aardvark here, if you look at this tree, is kind of the special branch here. It goes way back in time and doesn't have any other close relatives. So the notion is if we lost the aardvark, we'd be losing a huge chunk of evolutionary time. And there's nothing like an aardvark around to sort of take the place of it, so to speak. On the other hand, if we lost one of these mice up here, there's a bunch of mice, you know, so we're not losing a huge chunk of the tree of life. We're just using a part of it. So the idea is that's the intuition, and then you can actually put numbers on it. And there's a whole international program devoted to identifying which are the species that are most like the aardvark and which are the ones that are more like the mice. So the idea is we can actually put a score called an EDGE score on a species and say, you know, that one is a really special branch of the tree of life. So I'll give you an example. From the previous example, ginkgo is a good example. So, you know, here's our tree and here's ginkgo. If I had to choose one species here that I really wanted to save, it would be the ginkgo. And I would hate to make that choice. That would be a horrible choice. But if I was forced to, I'd say I'm going to save the ginkgo. And the reason for that is the ginkgo is a huge long branch. It's probably the oldest species on Earth. It goes back 170 million years. There's nothing like a ginkgo anymore in the world. And so if you lost that, you'd lose all of this knowledge of what happened all through that whole time period. That whole bit of evolutionary history would be totally gone. And it's really evolution, it's so distinctive, right? It's got these weird leaves. It's got incredibly weird seeds with this wonderful little gametophyte in the middle that we sometimes eat. It's chemically weird as can be, and that's why you find it as in drugstores as an extract. And it's extinct in the wild, essentially. This is the only populations that are thought to be wild populations. I mean, it's now grown around the world, but they're all kind of a monoculture. So in terms of actual standing genetic variation, it's just those few populations. So ginkgo is a good example of a high edge species. You know, you would definitely want to save that one. Another one from my own work actually is this one. I'll just mention it because I like it a lot. We discovered some years ago that when we did the phylogeny of all the flowering plants, there was one species called Ambarella that stood out as the sister group to all other flowering plants. So it's one species, and then all other flowering plants are 250,000 species. So, you know, this is a very special branch of the tree then. And it's a weird plant in so many ways that I can't go into. And it's also globally endangered. It's only in New Caledonia, and it's only these populations. And some of them are disappearing pretty quickly, as a matter of fact. So this was another high edge species. This is a weirdo thing, deep branch in the tree, right? And globally endangered. So that gives you the idea of how we could use phylogenies to help assess, you know, where we might want to focus some attention. We can do the same with areas. I just want to briefly mention this to you. So we can say, what area would we choose to save? Or how do we value different areas for conservation? Not just individual species, but particular regions of the globe. So let's suppose we have two areas. This is area 1. All the species in area 1 are all really closely related to one another. All the species in area 2 are distantly related. They have the same species diversity. They each have 4 species, but they're related in different ways, right? I mean, they're covering more or less of the phylogenetic tree. I think that you can see that pretty obviously. So that's the basis of a further evaluation of areas. So if I had one area and it had ginkgo and apple in it, and the other area just had the magnolia and the apple, I'd say I want to save area 1 first. That's going to be my priority area because it's got so much more of the tree of life is represented, so much more of evolutionary history is represented in that area. The same goes for what's called feature diversity. So when you have lots of evolutionary time, you're likely to evolve lots of interesting things, like along this branch, all the weird ginkgo chemistry and all the weird ginkgo morphology. So you can add up the amount of change along the branch, and this is an area that captures a lot more different features than this area over here. Right. So this is the way we can use phylogenies to assess areas and say, oh, that's an area that's got a lot of phylogenetic diversity. Now, this has been done on many scales. I mean, here's a very large scale thing. This is the country of China, of course. And what people have said about China, and I think rightly, is that this part of China is full of lots of deep branches in the phylogeny, very distinctive things and many things that are actually endangered. So there's a lot of biodiversity in this region that's sort of deep history. On the other hand, in contrast, the Tibetan Plateau is all these recently originating species. I mean, it's nothing deep at all. It's just lots of recently originating species. So we come to call these areas cradles of biodiversity because they're places where you're generating a lot of species. And these are called museums because you're sort of storing the old stuff in a sense in the museum, right? So that's the concept of cradle and museum. And this can be done on multiple scales. Here's for the Atlantic forest. These are amphibians. So Dave, you can relate to that. In the Atlantic forest of Brazil. And this is a map of the phylogenetic diversity on a pixel by pixel sort of basis, right? You can do this on any scale. And it says, look, the phylogenetic diversity is really in this area right here. It's not so great over here. And then you can predict into the future and say, well, you're going to lose a lot of phylogenetic diversity in this area here. So maybe that's an area we should concentrate some effort. Okay, so that's part 2. And that's sort of a segue to the last part, which is about the viburnum stuff. So there are methods to evaluate distinctiveness of species, the phylogenetic diversity of areas, and that can help us a little bit think about conservation. So I want to take that perspective and now actually apply it in an actual group of organisms. And that's the group that I study, this plant group, Viburnum. And it's about 163 species on any given day. I might change my mind a little bit on this, but it's around that, right? And they're distributed around the Northern Hemisphere. But importantly, they're here in Borneo in Southeast Asia, and they're here in the Andes here in South America, as I'll mention to you in a minute. And they have fleshy fruits that are dispersed by birds, various flowers, etc. But I'm not going to concentrate on any of that. I want to take what I just did about phylogeny and conservation and apply it to viburnum. That's what I'm about to do. So here's a phylogeny. Can you recognize it as it's like your fingers, but multiple fingers. So there's all these 163 tips in here. And it's dated phylogeny, so it has millions of years on this axis. And we can do that because we have some fossils we can stick in here to kind of calibrate it so we can sort of figure out the ages of things. So we have a pretty good understanding of the phylogeny and the way in which diversity has built up over time. And a lot of diversity has originated recently, just in the last 10, 12 million years, something like that. We can also, we can also say, how has viburnum moved around the world during its history? So we can reconstruct that. These are the areas where viburnum, this is a polar view of the Earth. These are the areas. Asia is the red area here. And we can reconstruct on this tree where things probably lived in the past. And so what we have learned from this is the viburnum probably lived in the Asian tropics or warm forests in Asia before they moved anywhere else. And then they moved out around the world several times. And just to make this simple, here's a different diagram. They originated over here maybe 70 million years ago, right around the KT boundary area, and then spread 6 times into the New World. And one of those lineages moved recently, in the last 10, 12 million years, down into South America. And that's going to become important to us, okay? So that's the history. And there's some interesting other changes that happen with the leaves as they evolve into these different areas. Now, take that edge approach that I talked about before. What viburnum species are on the really long branches that go way back in time and have no close relatives? So it turns out there's two of them, this one and this one. This one here goes back 60 million years, if we're correct about this, and it has no close relative on this branch. It's called clemensi. This one goes back maybe 50 million years, no close relative. So these are two things that they're the ginkgo equivalent in viburnum. These are like the ginkgos of viburnum, right? And where do they live? Both of them, it turns out, live here and only here, Mount Kinabalu in Borneo. Isn't that bizarre? So these two really long branches of evolutionary history are preserved in this one spot on Earth, Mount Kinabalu. I'll say more about each one of these now, just for a second, to make the point that they really are interesting and weird and endangered. And so this was described first by a person named Mary Clemens. I mean, she found it and collected it. This was her collection. It's a diagram of it. It had one fruit on it. That's all we know about it, basically, and some leaves. And then when I was there, I happened to be there stumbling around in 1993, and I ran into this damn thing, and I recognized it as a viburnum. It's like, and I was pretty much almost the second person to see this thing. And this is what I had at that time. I picked up a sprig of it, took a picture of it, and that was it. And there was nothing, you know, it's just some leaves. There were no flowers, no fruits. There was nothing known about it. And anyway, Erica Edwards and I, Erica's in the back of the room, took a trip over to Borneo to try to find this thing and look at it again. And we found it all right. That's me standing underneath a plant, extremely rare plant, actually. And we climb up the tree, and what did we get? One damn fruit. And so now you cut the fruit open, it's the weirdest viburnum fruit you'll ever see. I mean, I recognize it as not really viburnum-like at all. You guys wouldn't know, but I'm just telling you, it's astounding in its own weird way. But we still hadn't found the flowers of this thing. So we went back in 2014, you know, determined to try to find flowers of this thing. And we did eventually discover the flowers. And what was so weird about it is it's a dioecious plant. That is, it has males and females. And we were just seeing the female with the one fruit on it. We never saw any males, no sign of that. But it turns out this is a male flower and that's a female flower, and they're on different plants. Also, they're the only viburnums that have separate petals. All the rest are formed into a tube and everything. So this is, by any measure, it shouldn't even be a viburnum practically. It's the weirdest damn thing, right? So I'm saying it's an evolutionarily distinct and globally endangered species, for sure. The other one is amplificatum, also grows there on Mount Kinabalu. It's weird too. It's got the largest viburnum leaves. This is the size of most viburnum leaves. It's got the largest viburnum fruits that mature and they're yellow, and I think they're dispersed by orangutans. Bizarre, not birds, which are most. And then the flowers, which we were the first to discover, are weird too. I mean, they have really crinkly little stamens, and everything's wacky about this plant. So we got two super long branches existing side by side, sort of, in Mount Kinabalu. Now, what else is in Mount Kinabalu? Well, it turns out there are two other species that are right over here. And what I've done here is I've drawn with my little magic marker, I drew and connected them, right? So this shows all of the evolutionary history of viburnum that's preserved in Mount Kinabalu, in 4 species on Mount Kinabalu. A huge amount of viburnum diversity, evolutionary history is preserved there. Now, fortunately, Mount Kinabalu, many of you have been there, I think, probably, but here's Mount Kinabalu. This is at the northern tip of Borneo. And it is a park. And so hopefully these things are somewhat protected there, and maybe they're going to survive, but they have very small population sizes. This is just showing the other 2 species that live there. They're weird as can be too. So you got 4 really oddball species living in a very small little area on Earth and capturing a huge amount of viburnum evolution. Now I want to contrast that to this situation. So here's what we find in Latin America. So this is a complete opposite situation. Here we have 42 species, so that's a lot of viburnum species. They're very recent. All of them go back about 10, 12 million years or so. And they're all very similar to one another. So this Borneo captures a huge amount of evolutionary history. Here, there's a lot of species, but hardly any evolutionary history, so to speak, right? And as you would expect, oh, I should say, we've been funded for some time now by the NSF to study this. We're just about to publish all these exciting results. But we've had a blast going in the field and collecting all these things all up and down the Andes and so forth. And the collaborators here are Patrick Sweeney from the herbarium, Erica, who's a professor in EEB, Darren Eaton, who was a postdoc with us, who's now at Columbia. And so we spend a lot of time in the field collecting these things and getting their DNA and stuff. And here's a map just to show you the distribution, and these are the collections of Viburnum all up and down. They live in the mountains above 1,000 meters, so they're up in the cloud forest, so to speak, covered with clouds and everything. Very wet environment. And they're all very closely related and very similar to one another. And I just want to make that point with a couple slides, how similar they are. So if you look around the world at Viburnum, you can see some flower variation. Maybe not a huge amount, but there's a lot of different kinds of Viburnum flowers. Now, look at the Viburnum flowers from Latin America. I can't tell them apart. I mean, the flowers of these things are pretty much identical. So there's 42 species. They all have flowers that are pretty much the same. What about the fruits? Well, if you look around the Viburnum all around the world, they have reds and blues and yellows and all sorts of great colors. What do they look like in Latin America? They're all the same. So we got 42 species evolving kind of independently. They have the same kinds of flowers, the same kinds of fruits. So how can this be? Well, they do vary in some other features. So the feature that's most noticeable is the leaves. So some of them have big, huge leaves. These are all to scale. These are silhouettes of leaves. And some are big, and some, like that one up in the corner, are really small. And so you get a wide variety of shapes and sizes of leaves evolving in these 42 species. And it turns out that there's other variables too, like whether they're really hairy. Like this one is really hairy on the underside. This one's hairy on the underside. This one has teeth on the margins. This one doesn't. So there's some features that differ with the leaves, right? And that's why we can recognize these species at some level. But here's the cool thing. If you go to Oaxaca, let's say, you look around, you see these three Viburnums growing near one another. There they are. You can see what they look like. You go down to Chiapas, you see three species growing near one another. They're not the same species, but they look kind of similar in some ways. Like this one kind of looks like that one. This one kind of looks like that one, right? So different species in these different areas, but they look the same. You know, they've evolved the same leaf morphology. Now, here's the embarrassing part of this story, I guess. So when I was a grad student, I sort of noticed this pattern, and I said to myself, how could that have come about? You know, how did that evolve? And when I published my dissertation way back in 1982, I published this view, and that was that all the big hairy ones were related to one another, and all the little guys were related to one another. And if that were true, then they must have evolved, these types must have evolved, and then gotten around to every different mountain area, right? So you evolved the different types, and then they moved to all the different mountains. That was what I said. And well, anyway, it turns out to be the complete wrong answer to this. And this is the right answer, it turns out, which is kind of remarkable, which is that there was a Viburnum that spread down through all these mountains. And then within each particular mountainous region, you evolved the different forms over and over again. So you're rapidly evolving these different forms of leaves in each area, right? And it turns out then that this one, which looks like this one, is not closely related to it. Instead, it's closely related to this, right? And so forth. And you end up with a pretty remarkable pattern here. Now, this is from the paper that we're about to publish about this. This is a phylogeny, and what we can show, we can reconstruct that they moved from Mexico eventually down to Argentina. So there's a progressive movement down the mountains. That's one thing we can show. And then within each region, we can show that there's multiple species within Oaxaca, within Chiapas, et cetera, you know, within northern Colombia. Each region has several species in it. And then we can also show that they've evolved these leaf types over and over again. So if I go to any particular region like Oaxaca, I can see there's one of this type, one of this type, one of this type, and one of that type. There's 4 types of leaves that have evolved in Oaxaca, right? And this is repeated in different areas. So it's a really remarkable thing. It's called replicated radiation. There are some good examples in animals, but they're not so good in plants. And maybe this is the first example, actually, in plants. It leads to some remarkable cases of kind of parallel or convergent evolution. Like this one's from Oaxaca, but it looks awfully much like this one here. You couldn't really tell them apart, but in the phylogeny, they're really distantly related, so far as you can go with relationships. Now, one last thing I'll tell you is that why did they evolve these different leaf types, you may wonder? You know, like what's the difference between a big hairy one and a small one? Does that adapt them to some particular environment or what? So with Erica leading the charge on this, we we did a study of 3 species, a detailed study of 3 species living in Chiapas. This is the point localities of the plants that we were studying. And I'm not going to take you through the details of this, but we had these weather stations that we put in the woods with the plants, right? So we could follow on a daily basis what was happening. So for example, this is average daily leaf wetness. This is how much cloud cover is in the forest. And you can see this species, which is this one over here, the big one, it's always wet. It's perpetually wet through the whole year. These other ones are really dry in the winter. So what I'm saying is we can distinguish the niches of these species. They're evolving into slightly different environments that are adjacent to one another in each of these several different areas down through the mountains. So I just want to wrap that up and then wrap the whole thing up. So the summary of this is, if you think about it, what we've learned is that northern Borneo, right, is this museum place for viburnum. It's maintaining these dying embers of some distant evolutionary past. Whereas when they get over into the New World and go down into the Andes, that's a hotbed of speciation. That's where the action is. If we want to study speciation, that's ongoing. That's where we go and check it out. Whereas if we want to study extinction, we go to Borneo and study those plants. So both areas, I think, are important to conserve, but for different reasons, that we can now identify that, you know, those are really different phenomena that are going on in those areas. One last point I want to make is that Erica and I and others are trying to push this model of deeply understanding some groups of organisms to get this level of information, because it's hard to get. And it takes like a fairly long lifetime of work to get this level of knowledge. And we want the NSF and everything to fund these kind of deep dives. We're calling it kind of the model clade approach, and we're trying to push that. And we think that'll be successful. Okay, so I've reflected enough on biodiversity, and at this point, I just want to stop and thank everybody. I'm eternally grateful to this, by the way, is a picture we took with Ed and Sasha down at their house in Fort Worth when we went down to visit. Was it last year? I forget when. And there's Allison, of course, and Dave. I don't know why Dave is centered in the middle of this. Yeah. And there's a plant that has a story too, which is that's a long branch in the cactus phylogeny. So that doesn't look like a cactus. That's a very distinctive cactus. So I don't know if that's still living at your house. And they're back. All right. Erica is the world's expert on that plant. So we were sort of worshiping that at the time. Yeah, so we found viburnum on your property. Yeah. But anyway, basically, I want to thank the Peabody. You know, I had I still have an association, a very good association with the Peabody Museum, and it's meant so much to me over these years. I can't tell you. And IPBES, you know, I got to know IPBES more recently, but I really enjoyed that stint with IPBES, and I think we did some interesting things, and it was so much fun to get to know that whole crowd of people as well. And then just thanks to all the rest of you. I mean, you know, I've worked with almost every one of you in some capacity, and it's just been a blast. And I can't thank you enough for all the support you've given me over the years. So with that, let's call it a day. Thanks.
I know Michael would welcome questions.
Yeah.
Wow.
Sure, I'll ask one, Michael. Thank you. You know, E.O. Wilson has the Half Earth book, and there's the 30 by 30 initiative. Can you talk a little bit about sort of what is needed to preserve biota? Oh, there's also one other statistic, which I think we've lost half the species on Earth in terms of just the number of species in our generation. I don't know whether that's a good statistic or not, but could you talk a little bit about those efforts and what we might do and what we might be thinking about to try to preserve species on this planet?
Yeah. The last thing that you said, I don't recognize. That is to say, we haven't lost half the species on Earth over this time. We're on the brink of a lot of extinction. There's no question about it. We're experiencing a lot of it now. And it will get worse over the next couple of decades. Hopefully we'll wise up a little bit. And the rates of extinction now are much higher than they used to be. I mean, we can calculate that. So it's 10 to 100 times the background rate of extinction going on right now. But in terms of these conservation efforts, yeah, the 30 by 30 thing is a really important kind of effort that's getting off the ground now. A bunch of countries are now committed to that 30% I mean, the United States is trying to do its part, Canada, Mexico. There's a bunch of partners in this around the world that have committed to the 30% of land set aside. Unfortunately, a lot of it, okay, so that's a good thing. I mean, I'm not gonna, you can't run that down. That's a really good thing. We should set aside this kind of, and the half earth thing is kind of admirable idea also, I think. But, actually implementing these things and making them be meaningful is really difficult, and the set aside areas in some of the countries that are participating are not the areas that have the most biodiversity, for example, right? In other words, it's 30% of the land, but it's not necessarily, by the measures that I was showing you, it's not necessarily the most interesting bits of biodiversity. So there's a kind of a mismatch that goes on at that level. So we need to really be identifying where are the hotspots of biodiversity that we really should be focusing attention on and setting those aside. But then all of that has so many other, really important consequences and problems, for people who live in those areas, et cetera. So it's really a big deal and non-trivial how to set aside the right chunks of land. But the effort is admirable. And of course, in the end, that's what we need to be doing, right? If we want, I'm very excited about preserving various species, right? But the way to do the various species thing is to set aside the land, in other words. And I would motivate some of that effort along the lines of saving species of, like I was saying, right? But those are great efforts. Yeah, Peter.
I apologize if
So I should have said, yeah,
I apologize if this is naive. Are there politics between the idea of saving museums over cradles versus cradles over museums, or is it just kind of just accepted as two different interesting ways of carving up the species world?
Yeah, so I think initially the idea was we really should be, if we could identify things that we thought were museums. Okay, now here's a concept that you got to get your brain around. So I just identified what I consider to be a museum, namely North Borneo, for Viburnum. Now, would that expand to anything? Is there anything else in that same category? Is that a museum for everything else? And the answer is it's complicated, right? So not every group of organisms is going to have the same museums and cradles. So right off the bat, it gets complicated. But at first, I think people were saying, attaching sort of a value judgment to it. Like if we could identify areas that had lots of evolutionary history, we would want to preserve that over an area that was a cradle, which is an ongoing hotbed of speciation. I don't think that's quite the right attitude, because there are, I think there's distinctive things we could learn by segregating, by understanding that landscape. We could then focus and learn various things. Like I said, if we want to understand the genesis of biodiversity right now in certain groups, the Andes are going to be really important. And that's not just for Viburnum, it's for a lot of other groups too. So at first there was a value judgment and some politics surrounding that. But I think now people have backed off and said, well, these are really interesting distinctions. We have to take them on board, but it's not so cut and dried. Yeah. Yeah, John.
Mike, I was wondering if in the EDGE approach, if there's an additional factor that you might incorporate from your coffee example, where you might have groups that are closely related and species rich, and you might think are uninteresting. Maybe mice aren't interesting, but coffee could be.
Yeah.
Crop organisms, or
Right.
things like venomous marine life, or
Right.
that might be important for medical reasons. Right. Do you see people incorporating a numerical or quantitative approach to that too?
Yeah, well, so the way it's been done in the literature is mostly in a generic way with things like this feature diversity. There are measures of how distinctive things are in terms of just the number of changes. But what that doesn't incorporate is whether it's an interesting thing or not. So caffeine is definitely interesting, right? And so you could add that into the mix. It's like, we want to save the ones that have caffeine over the ones that don't, right? Yeah. Yeah, exactly. So in other words, that's another dimension to the problem, is that there's particular features that we care more about than others. And we can identify some of those. Some of them we can't identify because we don't know enough, right? We don't really know the chemistry of most organisms. So we can't say how distinctive they are or interesting to us. But for some things that we can, like naked mole rats, for the reason you said, would be great, But yes, so there are ways to integrate that, I think, but it's not been done very much, except in an ad hoc way. You know. Yeah.
Michael, that was great.
Thank you.
That was great. I was wondering, in the American setting, there's a lot of mapping going on to try to show resiliency. So soils, mountains, change in temperature and water and so on. And wow, what if these species start moving within these resilient areas? Do you give much credence to that? Is that helpful or useful? How does it fit into your thesis?
Yeah, I mean, I think so. I think it's super important. So it's super important to be aware that things are moving around, right? In other words, we can identify right now where, you know, we say this is an area where there's a lot of interesting stuff going on, but it's also the case that things are going to be moving in and out of that area pretty regularly as climates change and things like that, right? So that's somewhat problematical, right? Because it's a moving target in a way. It's not to say it shouldn't stop us from identifying particular areas for whatever reason and trying to preserve them. But it's not like for all time that's going to be a solution, because for all time, you know, things are going to be moving, right? So I think we have to take on board the idea that it's not a static planet and things aren't going to stick around in the same places where we thought they would be, you know. But I think the idea of trying to identify, I mean, the resilience thing is an important thing. But movement, I'm sort of a biogeography guy, right? And organism movement is a thing, and it's going to be important in the near future. You know, I mean, what's happening, you know, maple trees, sugar maples are moving north, you know, I mean, they're not going to be here in Connecticut very much longer. Who said that? Yeah, one more question.
One more question.
Yeah, James. I know, right? I don't think we have to worry about it, right? I mean, it's like not a problem. I mean, we are definitely not globally endangered. We're what? Oh, good. Okay, thanks. No, I mean, humans are an interesting case. We're not we are sort of distinctive in several ways, but not as distinctive as we think we are, I would say. And but we do have a few things like a bigger brain and so forth. Eric, am I right? Yeah. Yeah, but we're not globally endangered, so that's for sure. I don't think we need to worry.
That's an excellent place to end this part. So why don't we thank Michael again. And so we're going to head upstairs now, and Michael has organized this entire thing. So he is his own warm-up act, because Michael and David Heiser are going to be playing for us upstairs during the reception.
The last lecture I heard you give was 30 years ago.
Oh yeah. Oh my God.
And this one was just brilliant.
Uh,
It was completely